In 2005, a researcher from North Carolina dissolved a tyrannosaur bone in acid. And when the minerals disappeared, soft tissue remained inside, transparent, flexible, vessels that had held their shape for 68 million years. She showed this to her colleagues. Her colleagues said, "Contamination." She showed it again. Again they said, "Impossible." 20 years later, in April of 2026, a network of preserved blood vessels was found inside the largest known Tyrannosaurus rex, a specimen named Scotty. In January of 2025, original collagen was confirmed in a duck-billed dinosaur from South Dakota using 5 independent methods. What scientists called impossible keeps happening, again and again. And it changes everything we thought we knew about fossils. Before we go any further, please subscribe to this channel. Your support really helps us to continue this work. Thank you. There is a word that appears in every introductory paleontology textbook, in every museum placard, in every explanation ever given to a child pointing at a dinosaur skeleton and asking what it is made of. The word is mineralization. The process, as it has been taught for a century and a half, goes like this. An animal dies. It is buried in sediment quickly enough to avoid complete destruction by scavengers and decay. Groundwater percolates through the sediment and through the porous structure of the bone. Minerals dissolved in that water: silica, calcium carbonate, iron compounds, gradually replace the original biological material. Molecule by molecule, over thousands and then millions of years, the organic content of the bone is displaced by inorganic mineral. What remains is a faithful replica of the original structure, sometimes of extraordinary precision, preserving the architecture of the bone down to the microscopic level of individual cells. But the original biology, the proteins, the collagen, the lipids, the cellular machinery, is gone, destroyed, replaced. What you hold in your hand when you hold a fossil is a stone carving of a once living thing, not the thing itself. This is the standard model. It is taught in every university. It appears in every textbook. And it turns out to be, at minimum, significantly incomplete, because some fossils are not entirely stone. Some fossils, under specific preservation conditions that we are only beginning to understand, retain traces of their original biological molecules, not replicas, not mineral replacements. The original molecules, proteins that were synthesized inside a living animal tens of millions of years ago, structural compounds that were part of a functioning biological system long before the first human being existed. Organic chemistry that has survived conditions that every theoretical model of molecular degradation says should have destroyed it completely. Those molecules are there. We know they are there. And in the past 2 years, the scientific case for their presence has become, for the first time in a debate that has lasted 3 decades, essentially unassailable. To understand what this means, you need to understand what it does not mean. It does not mean Dinosaur DNA. The popular imagination, shaped largely by a certain film franchise involving amber and theme parks, leaps immediately to the possibility of genetic resurrection, of recovering the sequence information that would allow extinct animals to be reconstructed from their molecular blueprints. That possibility remains, for reasons we will discuss, firmly in the realm of fiction. DNA is a far more fragile molecule than the proteins we are discussing. Its degradation timeline, even under optimal preservation conditions, is measured in hundreds of thousands of years at most, a figure confirmed by the analysis of ancient DNA from Ice Age specimens, the oldest of which approach 1 million years but do not exceed it. 66 million years is 2 orders of magnitude beyond that limit. Dinosaur DNA is not coming back. But DNA is not the only biological molecule that carries information about a living organism. Proteins carry it too. And proteins, as it turns out, are considerably more durable than anyone expected. Specifically, structural proteins. The fibrous proteins that give biological tissues their mechanical properties, that form the scaffolding of bone and cartilage and connective tissue that persist in the extracellular matrix long after cells have died. Of these, the most relevant is collagen, the most abundant protein in vertebrate bodies, the primary organic component of bone. A molecule with a distinctive triple helix structure that makes it both physically tough and, under the right conditions, remarkably resistant to degradation. It is collagen that sits at the center of the molecular revolution we are about to examine. And the story of how we came to know it survives in fossils at all is one of the stranger episodes in the recent history of science. It begins in the summer of 2003 in the Badlands of eastern Montana at a site called the B-Rex locality in the Hell Creek Formation. A team from the Museum of the Rockies, led by the paleontologist Jack Horner, was excavating a large theropod dinosaur, a Tyrannosaurus rex, as it turned out, a specimen that would eventually be catalogued as MOR1125. The skeleton was substantial and well preserved, but its removal from the rock posed a logistical problem. The femur, the thigh bone, was too large to be airlifted out of the field in one piece. A decision was made to break it. The bone was fractured in the field, and when it broke, the interior of the cortical bone was exposed to examination for the first time. What the team saw was unexpected. The interior of the bone showed what appeared to be vascular canals, the channels through which blood vessels run in living bone that retained what looked like their original flexible character. The bone was handed to Mary Schweitzer, a molecular paleontologist at North Carolina State University who had been working on the question of organic preservation in fossils for years, and who had, in the course of that work, developed a reputation for finding results that her colleagues found alternately fascinating and deeply uncomfortable. Schweitzer's laboratory took the femur fragments and demineralized them, dissolved away the mineral component of the bone using a chelating agent called EDTA, which selectively removes calcium without attacking organic material. Under the standard model of fossilization, this process should have left nothing. The mineral is the fossil. Remove the mineral and you should be left with, at most, some residual sedimentary contamination. What Schweitzer found instead was a flexible, translucent matrix that retained the three-dimensional structure of the original bone. And within that matrix, there were vessels, transparent, branching, hollow tubes that could be manipulated with forceps, that behaved like soft tissue, that in every observable respect resembled the vascular channels of modern bone. She stretched them. They stretched. She released them. They returned to shape. The paper describing these findings was published in Science in March of 2005 under the title ___, quote, _0, ___. The response from the scientific community was, to put it mildly, skeptical. The dominant interpretation was contamination. Modern biofilm, bacterial colonies, or other organic material that had infiltrated the fossil during its 68 million years in the ground and been mistaken for original tissue. This was not an unreasonable position. The claim being made was extraordinary. Extraordinary claims, as the saying goes, require extraordinary evidence. And the evidence in 2005, while compelling to Schweitzer and her collaborators, was not sufficient to close the argument. It opened it. What followed was nearly 20 years of one of the most sustained and technically demanding debates in the history of paleontology. The central question: are the organic materials found in dinosaur fossils original to the animal, or are they modern contaminants of various kinds, was examined from every possible angle with increasingly sophisticated analytical tools by researchers who held every possible position on the spectrum, from enthusiastic confirmation to resolute skepticism. The debate generated dozens of papers, multiple technical controversies about methodology, and a level of personal and professional investment on multiple sides that made it one of the more contentious ongoing arguments in the earth sciences. Through all of it, Schweitzer continued working, and the evidence continued accumulating. In 2007, Schweitzer's group reported the identification of collagen peptides from the same T-Rex specimen using mass spectrometry. The analytical technique that identifies molecules by their mass-to-charge ratios with a precision that allows individual amino acids to be distinguished. The collagen sequences recovered matched those of modern birds, specifically chickens and ostriches, more closely than they matched those of any other living animal group. This was exactly what evolutionary theory predicted. Birds are the living descendants of theropod dinosaurs, and their collagen should be most similar to dinosaur collagen. But the confirmation of that prediction through molecular rather than morphological data was a different kind of evidence entirely. It was, potentially, the first direct molecular confirmation of the evolutionary relationship between dinosaurs and birds. But the skeptics were not finished. The mass spectrometry data, while impressive, was challenged on grounds of contamination methodology. The collagen sequences identified were short, fragments rather than complete molecules, and the possibility that they represented contamination from modern bird or reptile sources that had entered the sample during preparation could not, the critics argued, be definitively excluded. The debate continued. New specimens were examined. new analytical protocols were developed specifically to address the contamination objections. And gradually, painstakingly, the picture began to clarify. The mechanism question was crucial. If original organic molecules could survive 68 million years in a fossil, there had to be a physical and chemical explanation for how that survival was possible. The biochemistry of protein degradation is well understood, and by every standard calculation, collagen should not survive on these timescales. At ambient temperatures, the half-life of collagen is measured in thousands to tens of thousands of years, not millions. Something had to be slowing the degradation dramatically. In 2013, Schweitzer's group proposed an answer: iron. The Hell Creek Formation, where the most significant soft tissue-bearing dinosaur fossils have been found, is notable for its exceptional preservation quality, but also for the high iron content of its depositional environment. When a dinosaur died and was buried in the Hell Creek sediments, its body contained large quantities of iron-rich blood. As the soft tissues decayed, this iron was released into the surrounding bone tissue. Iron, in the presence of oxygen, undergoes a reaction called Fenton chemistry, producing free radicals that cross-link nearby organic molecules, essentially tanning them in the way that leather is tanned, creating a chemically stabilized network of cross-linked proteins that is far more resistant to further degradation than the original uncross-linked molecules. On this model, the dinosaur's own blood preserved its tissues, creating a natural fixative that operated on a geological timescale. The iron hypothesis was testable, and it was tested. Blood vessels from modern ostriches were incubated in solutions containing hemoglobin, the iron-rich protein that carries oxygen in blood, and left for 2 years. At the end of 2 years, the vessels showed no signs of degradation. Control vessels without hemoglobin had decayed completely. The experiment did not prove that the mechanism operated in the Hell Creek dinosaurs, but it demonstrated that the mechanism could operate, that iron-mediated cross-linking could, in principle, preserve soft tissue structures on timescales far beyond what the standard degradation models predicted. This was the state of the science as it entered its 3rd decade of controversy. Compelling evidence, plausible mechanism, persistent methodological objections, no consensus. And then, within the span of a few months in 2025 and 2026, 2 studies were published that changed the character of the debate in a fundamental way. The first of these was the Edmontosaurus study, published in the journal Analytical Chemistry in January of 2025, by a team led by Professor Stephen Taylor of the University of Liverpool's Mass Spectrometry Research Group. The specimen at the center of this study was an Edmontosaurus sacrum, part of the hip region of a duck-billed hadrosaur dinosaur, an animal that lived alongside Tyrannosaurus rex in the Hell Creek ecosystem of what is now South Dakota, approximately 66 million years ago. The fossil weighed roughly 50 pounds, was part of the University of Liverpool's paleontological collections, and was considered exceptionally well preserved by the standards of the Hell Creek Formation. What the Taylor Group did with it was methodologically unprecedented. Previous studies of organic preservation in dinosaur fossils had typically relied on one or two analytical techniques, most commonly mass spectrometry for protein identification and immunological assays using antibodies against modern collagen as a test for the presence of collagen-like molecules. The critics of these studies had argued, with some justification, that no single technique was sufficient to exclude all possible contamination scenarios, and that the convergence of evidence required to establish the endogenous origin of any putative organic material needed to be more rigorous than the existing studies had achieved. The Taylor Group took this criticism seriously and responded to it directly. Their study applied not one or two, but a combination of independent analytical methods to the same specimen, with each method targeting different aspects of the putative organic material and each providing a different line of evidence for or against endogenous origin. The methods included cross-polarized light microscopy, which detects the optical property called birefringence that is characteristic of the collagen triple helix structure and distinguishes it from other organic molecules with different structural arrangements. It included tandem liquid chromatography, mass spectrometry, the most sensitive and specific form of mass spectrometry currently available for protein analysis, applied both to intact bone samples and to acid-digested samples from which the mineral component had been removed. And it included, critically, the targeted search for hydroxyproline. Hydroxyproline is an amino acid that is rare in biology generally, but is present in collagen in unusually high concentrations. It constitutes roughly 14% of all amino acids in vertebrate collagen, forming a key component of the triple helix structure that gives the molecule its mechanical properties. It is not significantly present in bacterial proteins, fungal proteins, or most other potential contaminating biological sources. Its presence in a sample is, therefore, among the most specific possible indicators of collagen. The Taylor Group found it, unambiguously, in multiple independent measurements, at concentrations consistent with the presence of genuine collagen. The paper's conclusion was direct. This research shows beyond doubt that organic biomolecules, such as proteins like collagen, appear to be present in some fossils. Not probably present. Not possibly present. Present. 30 years of debate, addressed by a combination of methods specifically designed to answer every major objection that debate had produced. The finding was not that all fossils contain organic molecules. The preservation conditions required appear to be exceptional and specific, and the vast majority of fossils are indeed fully mineralized in the conventional sense. The finding was that some fossils, under some conditions, retain original organic material. The exceptional becomes documented. The impossible becomes confirmed. The question shifts from whether to how. And then, 3 months later, April of 2026, the second study arrived. Scotty is not an ordinary T-Rex. Housed at the Royal Saskatchewan Museum in Canada, Scotty is the largest Tyrannosaurus rex ever discovered. Larger than Sue, larger than Stan, the most massive individual of the most famous dinosaur species in the history of paleontology. Scotty's skeleton shows the record of a life that was, by dinosaur standards, eventful. Multiple bones show evidence of injuries, healed infections, the kind of physical history that accumulates only in an animal that lived long and survived things that killed others. One rib in particular stands out, a large fracture, only partially healed at the time of death. A wound that the animal apparently carried for the last portion of its life without fully recovering from it. It was this fractured rib that became the focus of the 2026 study published in Scientific Reports. The research team, led by a doctoral researcher at the University of Regina, who had begun this work as an undergraduate physics student using particle accelerators to study fossils, applied synchrotron X-ray imaging to the interior of the fractured rib. Synchrotron X-rays produced by electrons traveling at nearly the speed of light in a particle accelerator are orders of magnitude more powerful and more precisely controllable than conventional X-rays, and they can be used to image the internal structure of dense materials, including fossilized bone, at a resolution that conventional imaging cannot approach. What the imaging revealed in Scotty's fractured rib were structures consistent with preserved blood vessels, specifically the iron-rich vascular structures associated with the bone healing response. In living animals, when a bone is fractured, the body responds by dramatically increasing local blood flow to the injury site, mobilizing the cellular and molecular machinery of bone repair. The bone around the fracture becomes temporarily hypervascular, densely supplied with new blood vessels carrying the materials needed for regeneration. These vessels leave microscopic traces in the bone tissue that can, under normal circumstances, be detected in histological sections of modern bone. In Scotty's rib, the synchrotron imaging found structures that appear to be the iron-rich remnants of exactly this healing vasculature. Blood vessels that formed 66 million years ago in response to an injury, and that have survived in mineralized and iron cross-linked form to be detected by a particle accelerator in a Canadian laboratory in 2026. The convergence of these two findings, the Edmontosaurus collagen, confirmed by mass spectrometry, and the Scotty blood vessels revealed by synchrotron imaging, in the space of 15 months represents something qualitatively different from the previous 20 years of incremental evidence accumulation. It is not that the earlier evidence was wrong. It is that the earlier evidence was always vulnerable to the methodological objections of the skeptics. These two studies were designed specifically to be invulnerable to those objections. They used different specimens, different institutions, different analytical approaches, different research teams with no shared personnel or institutional interests. And they reached the same conclusion. Original organic material survives in these fossils. The implications of this for paleontology as a discipline are not trivial. They begin with what we can now do that we could not do before. The most immediate application is in phylogenetics, the reconstruction of evolutionary relationships between extinct and living animals. The standard tools of phylogenetics are morphological. You compare the shapes of bones, the arrangements of anatomical features, the presence and absence of specific structures, and from those comparisons, you infer the tree of life. Morphological phylogenetics is powerful but has limitations. Convergent evolution, the independent evolution of similar features in unrelated lineages in response to similar selective pressures, can produce morphological similarities that are misleading about actual evolutionary relationships. And in deep time, where the fossil record is incomplete and the morphological distance between extinct groups and their nearest living relatives is large, the uncertainties compound. Molecular phylogenetics, using the sequences of proteins, or DNA, provides an independent and, in many respects, more reliable signal. The sequence of a protein is determined by the sequence of the gene that encodes it, and gene sequences evolve in a way that is more clock-like and less susceptible to convergent evolution than morphological features. If you can recover protein sequences from extinct organisms, you can place those organisms in the tree of life with a precision that morphology alone cannot achieve. This is exactly what the collagen sequences recovered from the T-Rex specimen in 2007 allowed Schweitzer's group to do, and the result, that dinosaur collagen is most similar to modern bird collagen, was molecular confirmation of what morphology had been suggesting for decades. With the methods now available, the tandem mass spectrometry and the rigorous contamination controls demonstrated in the Edmontosaurus study, the recovery of meaningful collagen sequence data from well-preserved Mesozoic fossils becomes a realistic research program rather than an occasional lucky result. Which dinosaur lineages had which collagen variants? How the structural proteins of the major dinosaur groups compare to those of modern birds and crocodilians, their nearest living relatives. What the molecular evolution of the vertebrate extracellular matrix looked like across the Mesozoic. These are questions that, a decade ago, would have been considered permanently unanswerable. They are not permanently unanswerable. They are answerable by the same methods that answered the Edmontosaurus question. We simply need more well-preserved specimens and more laboratories with the equipment to analyze them. Beyond collagen, there is the question of what other molecules might survive. Collagen is in some respects the ideal candidate for preservation. Its triple helix structure makes it physically tough. Its high hydroxyproline content makes it chemically distinctive. And its abundance in bone means it is present in large quantities relative to other proteins. But it is not the only extracellular matrix protein, and there is no a priori reason to believe it is the only one that can survive under exceptional preservation conditions. Elastin, fibronectin, osteocalcin, other structural proteins of bone and connective tissue, may also be present in well-preserved fossils, and their sequences carry evolutionary information just as collagen does. The systematic search for these molecules in the best preserved specimens from the most favorable depositional environments is now a legitimate and funded research program, not a fringe pursuit. And then there is the question that everyone asks, because it is the question that the popular imagination will not let go of, even though the scientific answer to it is clear. The DNA question. Can organic preservation extend to nucleic acids? Can any DNA survive on the timescales we are discussing? The answer, based on everything we currently know, is almost certainly no. The degradation of DNA is a fundamentally different process from the degradation of proteins, and it proceeds on a fundamentally shorter timescale. DNA is a relatively unstable molecule under geological conditions. Its backbone is susceptible to hydrolysis, chemical cleavage by water, and to oxidative damage. And the double helix structure that gives it its biological function is maintained by relatively weak hydrogen bonds that break readily at ambient temperatures. The oldest recovered ancient DNA currently comes from permafrost specimens approximately 1 million years old. And even this represents exceptional preservation at very low temperatures, well below the ambient conditions of the Hell Creek Formation. At the temperatures and chemical conditions under which the Hell Creek dinosaurs were buried and preserved, DNA degradation would have proceeded far faster than in Arctic permafrost. There is no plausible physical mechanism by which intact DNA could survive 66 million years in sedimentary rock. This does not mean that no information about dinosaur genomes is recoverable. The amino acid sequences of proteins carry indirect information about the DNA sequences that encoded them, because the genetic code specifies which codons, 3 nucleotide sequences in DNA, correspond to which amino acids. From a protein sequence, you can infer the possible DNA sequences that could have produced it, with some ambiguity at positions where multiple codons encode the same amino acid. This is not the same as recovering actual genomic DNA, and it does not allow the kind of complete genomic reconstruction that popular imagination envisions. But it is not nothing. As more protein sequences are recovered from more dinosaur specimens, and as the comparative genomics of living birds and crocodilians becomes more complete, it becomes possible to make increasingly detailed inferences about what dinosaur genomes might have looked like, not from the fossils themselves, but from the molecular evolutionary record preserved in their living relatives and cross-referenced with the direct protein data from the fossils. There is one more dimension of this story that deserves attention, because it speaks to something larger than the technical details of mass spectrometry and protein preservation. It concerns the sociology of science, the way that scientific communities respond to findings that challenge established consensus, and what the 20-year history of the soft tissue debate tells us about both the strengths and the limitations of that response. Mary Schweitzer's initial findings in 2005 were met with skepticism, and that skepticism was, in the specific context of what was known at the time, entirely rational. The standard model of fossilization was well established, well supported by decades of observation and experiment, and the claim that soft tissues could survive on geological timescales contradicted it in a fundamental way. Scientific skepticism toward extraordinary claims is not a failure of imagination. It is a necessary feature of a system designed to distinguish reliable knowledge from interesting speculation. The critics who challenged Schweitzer's methodology were doing what good scientists are supposed to do. And Schweitzer's response: to take the criticism seriously, to develop better methods, to seek independent replication, to engage directly with every specific objection rather than dismissing them, was equally exemplary. The 20-year debate was not a dysfunction of the scientific system. It was the scientific system working as designed, slowly and sometimes frustratingly, toward a conclusion that could withstand scrutiny from every direction. The conclusion, now available in 2026, is that the scrutiny has been withstood. The organic molecules are there. The methods for detecting them are rigorous. The contamination objections have been addressed. What began as an impossible observation has become a confirmed phenomenon, with a plausible mechanism, a growing body of replicating evidence, and an emerging research program built on its implications. This is how science makes progress, not through sudden revelation, but through the slow, contested, methodologically demanding process of building a case that the community of skeptics can eventually no longer reasonably reject. And the progress that now becomes possible because of that process having been completed is extraordinary. Think about what the molecular preservation of dinosaur proteins actually means for our understanding of these animals. For a century and a half, everything we knew about dinosaurs came from their bones. Bones tell us enormous amounts: body size, locomotor biomechanics, growth rates, diet, in some cases even behavior. But bones are the hard parts. They are the durable residue of an organism that was mostly soft tissue, mostly biochemistry, mostly the kind of biological complexity that bones alone cannot capture. We have known the skeleton of T-Rex in remarkable detail for decades. We have had extensive debates about its coloring, its social behavior, its thermoregulation, its metabolism. But these debates have been constrained by the limits of what bones can tell us. They have been, in a fundamental sense, debates about shadows. The animal itself, its proteins, its biochemistry, its molecular relationship to the living birds that are its descendants, has been inaccessible. It is becoming less inaccessible. Not quickly, not completely, and with many uncertainties that will take decades to resolve. But the direction of travel is clear. The molecular paleontology that Schweitzer began with a dissolving bone in a North Carolina laboratory 20 years ago, and that the Liverpool and Regina teams have now advanced to a level of methodological rigor that ends the debate about whether the molecules are real, is going to tell us things about dinosaurs that we could not otherwise know. Things about how their proteins evolved, things about the molecular basis of their extraordinary success as a group for 135 million years. Things the bones, for all their precision, were never equipped to tell. Now let me tell you what I think about this, as someone who has followed this story through its full arc. 20 years of criticism made the eventual confirmation stronger than any quick acceptance could have. Schweitzer didn't win the argument. She outlasted it. And what she produced in the process is not just a confirmed finding. It is a methodology that the next generation of researchers can trust. There is also something I find philosophically striking about what these findings mean for the concept of deep time. We talk about geological time constantly in this channel. the hundreds of millions of years, the events that happened before our. Anything we can relate to existed, and one of the persistent challenges of communicating deep time is the difficulty of making it feel real rather than abstract. 66 million years is a number. It is a large number, but numbers of that magnitude do not land with the same weight as something you can touch. Mary Schweitzer touched it. She held in her hands, stretched between forceps, a vessel that had carried blood inside a living animal 68 million years ago. That is not an abstraction. That is the past made physically present in a way that no number can replicate. The molecular record of life is older and more complete than we knew. It is preserved not only in the DNA of living organisms, which encodes the evolutionary history of every lineage in its sequence, but in the proteins of dead ones. Locked inside the mineral matrix of fossils that we have been treating as stone for 150 years, we are only beginning to understand how to read that record. The tools are now good enough. The debate about whether the record exists has been settled. What remains is the extraordinary work of actually reading it, of building, molecule by molecule, protein by protein, specimen by specimen, a biochemical portrait of the lost world that the bones alone could never fully render. The fossils are not silent. They never were. We just did not yet know how to listen. Понял? Это справедливо. Моя ошибка. Я должен был сразу писать полный сценарий на 90 минут, а не заставлять тебя каждый раз просить расширения. Это твое время и твои нервы. Исправляюсь. Пишу все 5 блоков расширения прямо сейчас. Чистый текст для диктора. Продолжение сценария с того места, где он закончился. Без заголовков и меток. The story of organic preservation in fossils does not begin and end with the Hell Creek Formation of South Dakota and Montana. It is a story that has been assembling itself from multiple directions simultaneously in multiple parts of the world, across different geological periods and different preservation environments. The Hell Creek findings are the most extensively studied and most rigorously documented for reasons that have as much to do with the extraordinary concentration of paleontological resources in North America as with any unique property of the formation itself. But the phenomenon is not local, and understanding its full geographic and temporal extent is essential to understanding what it actually means. Consider the amber record. Amber, fossilized tree resin, is among the most exceptional preservation media known to paleontology. It encases organisms that fall into or become trapped in fresh resin, sealing them from oxygen, from water, from the bacterial and fungal decomposers that normally reduce organic material to its component elements within years or decades. The result is preservation of soft tissue detail that no other fossilization process approaches. Insects preserved in Baltic amber from 30 to 40 million years ago retain the microscopic structure of their compound eyes, the fine setae on their legs, the venation of their wings, in a state of preservation that would be extraordinary in a museum specimen collected last year. The physical structure of these organisms is present in a fidelity that borders on the uncanny. But physical structure is not the same as molecular preservation. The question of whether the organic molecules inside amber-preserved organisms are original Whether the proteins and lipids and nucleic acids of a 30-million-year-old insect are still present in chemically intact form has been asked repeatedly and answered with increasing precision over the past 3 decades. The picture that has emerged is complex. Lipids, fats, and oils appear to survive in amber specimens with considerable fidelity, detectable by techniques like gas chromatography, mass spectrometry, and showing chemical signatures consistent with their biological origin. Proteins are more variable. Some amber specimens show evidence of protein-derived molecules. Others show complete replacement of original proteins by amber-derived organic compounds. DNA, despite early claims to the contrary that inspired a certain well-known franchise, does not survive in amber on the timescales that make headlines. The oldest credible ancient DNA from amber specimens is measured in tens of thousands of years, not millions. The claims of Cretaceous insect DNA from amber published in the 1990s have not been replicated and are now widely considered to have involved modern contamination. But the amber record demonstrates something important. The preservation of organic molecules in fossils is not a unique quirk of the Hell Creek iron chemistry. It is a property that emerges in different forms and with different degrees of fidelity from multiple preservation environments. The common thread appears to be rapid exclusion from the conditions that drive organic degradation: oxygen, water, and microbial activity. Amber achieves this through resin chemistry. The Hell Creek Formation achieves it through rapid burial in iron-rich sediment and the subsequent cross-linking of organic molecules by iron-mediated Fenton chemistry. Other environments achieve it through other means. Permafrost achieves it through cold. Exceptional burial in anoxic, hypersaline somnico, hoambosin, or otherwise antimicrobial sediments achieves it through chemistry. The principle is consistent even when the mechanism varies. This matters because it suggests that the search for organic preservation in fossils should not be limited to the Hell Creek Formation, or to North American Cretaceous deposits, or even to dinosaurs. It should extend to any well-preserved fossil from any depositional environment that provided the conditions for molecular stabilization. And indeed, when researchers have looked systematically in such environments, they have found organic material. Melanosomes, organelles responsible for the production and storage of pigment, have been identified in the feathers of Mesozoic birds and non-avian dinosaurs from Liaoning Province in China, preserved in a form that has allowed researchers to infer the original coloration of animals that died 120 million years ago. The iconic feathered dinosaur Anchiornis, known from dozens of specimens from the Tiaojishan Formation, has been the subject of melanosomes analysis that suggests a color pattern of black and white with a reddish crest, a level of biological detail about a Jurassic animal that, a generation ago, would have been considered permanently beyond the reach of science. The melanosomes work is worth examining in some detail because it illustrates both the power and the methodological challenges of molecular paleontology in a particularly clear way. Melanosomes are tiny organelles, roughly a micron in length, that contain the pigment melanin. They come in two basic shapes: spherical eumelanosomes that produce black and gray pigments, and elongated phaeomelanosomes that produce reddish and brownish pigments. In living birds, the distribution and relative abundance of these two melanosome types in the feather microstructure determines color pattern with a high degree of predictability. If you know the melanosome geometry and distribution in a feather, you can infer the color of that feather with reasonable confidence. The key discovery, made independently by several research groups in the late 2000s and early 2010s, was that melanosomes preserve in fossilized feathers. The organic material of the feather itself may be gone, replaced by a carbonaceous film or by mineral compounds, but the melanosomes, with their dense melanin content and their robust physical structure, survive as physical objects embedded in the fossil matrix. They are identifiable under scanning electron microscopy by their distinctive shapes and sizes, and their distribution in a fossil feather can be mapped with the same techniques used to map melanosomes in modern feathers. The result of applying these techniques to well-preserved feathered dinosaur fossils from China has been a growing catalogue of color reconstructions for Mesozoic animals. Sinosauropteryx, one of the first feathered dinosaurs described from Liaoning in 1996, had a striped tail of alternating reddish-brown and pale bands. Microraptor, the four-winged dromaeosaurid, had iridescent black plumage consistent with the eumelanosome distribution seen in modern starlings. Kaihongjuji, a Jurassic bird-like dinosaur described in 2018, had iridescent rainbow-colored head feathers. The structural coloration produced by the specific nanostructural arrangement of melanosomes in its feather barbules. These are not speculative reconstructions. They are inferences from physical evidence made by the same kind of structural analysis that allows color inference in modern feathers, applied to fossils that preserved the relevant structures through 100 million years of geological time. The coloration work represents a different kind of molecular preservation from the collagen story. It is primarily physical rather than chemical, resting on the preserved structure of melanosomes, rather than on the detection of specific protein sequences. But it is part of the same broader phenomenon, the survival of biological information in fossils beyond what the standard model of mineralization would predict. And it raises the same question, posed in a different key. What else survives that we have not yet looked for? The answer increasingly appears to be quite a lot. Research published in the past decade has reported the detection of original lipids in fossil specimens, ranging from Cretaceous fish to Eocene insects to Miocene marine invertebrates. These lipid signatures, detectable by gas chromatography, mass spectrometry, and related techniques, carry information about the metabolic biochemistry of the organisms that produced them, information that bone morphology cannot provide. The types of lipids present in an organism reflect its diet, its thermoregulatory strategy, its taxonomic position. In some cases, lipid analysis of fossil specimens has been used to make inferences about whether an extinct animal was endothermic or ectothermic, whether it generated its own body heat or relied on external sources. A question that for dinosaurs specifically has been one of the most contentious in the field for decades. There is also the emerging field of ancient pigments beyond melanosomes. Porphyrins, the pigment molecules that give blood its red color and chlorophyll its green, have been reported in fossil specimens of exceptional preservation. In 2018, a study of fossil ink sacs from Jurassic cephalopods described the preservation of the original melanin pigment in a form that was chemically essentially identical to the melanin in the ink sacs of modern cuttlefish. The molecule was 160 million years old. It was still recognizably, chemically, what it had been when the animal was alive. The researchers who described it noted that they had used the fossil ink to make a drawing, that they had literally written with pigment that a Jurassic cephalopod had synthesized. The image of that act is worth sitting with for a moment. 160 million years of geological time, and the molecule still worked. Now we need to talk about what all of this means for the specific question of dinosaur biology. The question that, for most people encountering this science for the first time, is the most immediately interesting one. What can molecular preservation tell us about dinosaurs specifically that bones alone cannot? The collagen sequence data is the most directly informative, and the implications of what has been recovered so far are significant. The collagen sequences identified by Schweitzer's group from the 2003 T-Rex specimen, published in 2007, showed the greatest similarity to the collagens of modern chickens and ostriches, both birds, and therefore both members of the avian dinosaur lineage. This result was consistent with the morphological evidence for the evolutionary relationship between theropod dinosaurs and birds, but it provided independent molecular confirmation of that relationship, a fundamentally different kind of evidence that cross-validates the morphological phylogenetics. When two independent lines of evidence converge on the same conclusion, the confidence in that conclusion increases in a way that neither line of evidence alone could achieve. But the collagen data can go further than simply confirming what morphology already suggested. Collagen sequences vary between lineages in ways that reflect their evolutionary divergence, and those variations carry information about the timing and pattern of evolutionary events. With sufficient collagen sequence data from multiple dinosaur groups, from theropods, from sauropods, from ornithischians, from the various lineages within each of those major clades, it becomes possible to build a molecular phylogeny of dinosaurs that is independent of the morphological phylogeny and can be compared to it. Where the two phylogenies agree, confidence is reinforced. Where they disagree, there is something interesting to investigate. A possible case of morphological convergence that misled the bone-based analysis, or a possible artifact of the molecular data that needs to be examined more carefully. This program is in its early stages. The number of dinosaur specimens that have yielded usable collagen sequence data is still small. A handful of T-Rex specimens, a few hadrosaurs, some fragmentary data from other groups. But the methodological advances represented by the Edmontosaurus study of 2025 mean that the recovery of collagen sequences from well-preserved Mesozoic fossils is now a reproducible procedure rather than an occasional lucky result. The systematic application of these methods to the best preserved specimens in museum collections around the world, and there are thousands of such specimens, most of which have never been subjected to molecular analysis, is now a defined research program with a clear methodology and a growing community of practitioners. Beyond collagen, the thermoregulation question deserves particular attention because it has been so central to the popular understanding of dinosaurs, and because molecular evidence has the potential to address it in ways that bones alone cannot. The question is whether non-avian dinosaurs were endothermic, warm-blooded, generating their own body heat through metabolic processes as birds and mammals do, or ectothermic, cold-blooded, relying on external heat sources as lizards and crocodilians do. The debate has evolved considerably over the past 50 years, and the current consensus, informed by bone histology and other lines of evidence, is that most non-avian dinosaurs were neither fully endothermic nor fully ectothermic in the modern sense, but occupied an intermediate metabolic position sometimes called mesothermy, with metabolic rates higher than living reptiles but lower than living birds and mammals. The details, however, remain contested. Lipid analysis of dinosaur fossils has the potential to contribute evidence to this debate. The types and ratios of fatty acids produced by an organism reflect its metabolic strategy. Endotherms and ectotherms produce characteristically different lipid profiles in ways that are detectable by mass spectrometry. If lipids can be recovered from dinosaur bone in sufficiently intact form, a significant if, but one that the existing evidence for organic preservation makes worth investigating, they could provide direct biochemical evidence about metabolic rates in specific lineages. This is speculative at the current state of the science, but it is not wildly speculative. It is the kind of inference that the new methodology makes worth attempting. There is also the question of immune system proteins, growth factors, and the other signaling molecules that are present in bone tissue, and that carry information about the physiology and life history of the organism that produced them. Osteocalcin, a bone-specific protein involved in bone mineralization and calcium regulation, has been reported in dinosaur specimens, and has been the subject of analysis suggesting that dinosaur bone physiology resembled that of modern birds more closely than that of modern reptiles. Vascular endothelial growth factor, which plays a role in blood vessel formation, has been reported in some specimens. These findings are preliminary and remain to be independently replicated with the kind of methodological rigor that the Edmontosaurus collagen study achieved, but they point toward a future in which the biochemical portrait of a dinosaur extends well beyond its structural proteins to include the signaling molecules that regulated its growth, its reproduction, and its response to injury and disease. This last point connects directly back to Scotty and the fractured rib that was the focus of the 2026 synchrotron study. The reason that Scotty's injury site was specifically interesting for molecular analysis is that bone healing is one of the most biochemically active processes in vertebrate physiology. A healing fracture is a site of intense cellular activity, of growth factor signaling, of vascular proliferation, of osteoblast and osteoclast activity, of collagen synthesis and remodeling, all of which leaves molecular traces in the bone tissue that are detectable, if anything survives, at a higher concentration than in uninjured bone. Scotty's rib, in other words, was not just a record of an injury. It was, potentially, a record of a biological response to that injury, a molecular snapshot of a living process that occurred 66 million years ago and left its chemical signature in the iron-rich sediment of the Hell Creek Formation. The synchrotron imaging showed the vascular structures. The next step, already under investigation by the research team, is to combine the imaging data with mass spectrometric analysis of the same tissue to determine whether the molecular signatures of the healing response are detectable alongside the physical structures. This is the frontier, not the confirmation that organic material survives, which is now established, but the determination of what specific biological information that organic material contains, and how completely and faithfully it records the biology of the living animal. What this means for the next 20 years of paleontology is not difficult to project. The techniques exist. The methodological framework exists. The institutional infrastructure, the particle accelerators, the mass spectrometry facilities, the paleoproteomics research groups in universities on multiple continents exists. What is needed now is the systematic application of these tools to the most promising specimens in the world's major natural history collections, most of which have never been subjected to molecular analysis. Because, until very recently, there was no established methodology for doing so that could withstand scientific scrutiny. The Natural History Museum in London holds hundreds of thousands of vertebrate fossil specimens. The American Museum of Natural History in New York holds comparable numbers. The Smithsonian, the Field Museum, the Museum of Natural History in Paris, the collections of Chinese institutions that hold the extraordinary Liaoning feathered dinosaur specimens. Together, these repositories constitute a molecular archive of incalculable scientific value that has been sitting largely unread for a century and a half, waiting for the methods that would allow it to be read. Those methods now exist. The reading has begun. It will not happen quickly. Each specimen requires careful evaluation of preservation quality before molecular analysis is attempted. The analytical procedures are time-consuming and expensive. The interpretation of results requires expertise in paleontology, biochemistry, and mass spectrometry that rarely coexists in a single laboratory, and requires collaboration across disciplinary boundaries that scientific institutions are not always well designed to facilitate. The career incentives of academic science do not always reward the patient. Methodologically demanding work of building a molecular database, one specimen at a time, when the rewards of academic recognition tend to flow toward the discovery of new species and the description of spectacular specimens. These are real obstacles, but they are not insurmountable, and the direction is clear. Every year, more specimens will be analyzed. More protein sequences will be recovered. More color patterns will be reconstructed. more biochemical windows into the lives of extinct animals will be opened. The fossil record that seemed, for a century and a half, to be a record purely of form, of shape and structure, of the hard architecture of extinct organisms stripped of everything soft and living, is revealing itself to be something considerably richer. It is a record of chemistry as well as form, of biology as well as geology, of life, in some sense, as well as death. Mary Schweitzer, in an interview given not long after the 2005 publication, described the moment she first saw the flexible vessels under her microscope. She said, and I am paraphrasing from memory, that she found herself thinking about the animal, not about the fossil, about the animal that had lived, that had moved through a world 68 million years before her, that had a biology, a functioning, warm, metabolically active biology, and that had left, in the mineral matrix of its preserved bone, a trace of that biology that had waited in the dark for her to find it. She said it made the animal feel real in a way that the skeleton alone never quite had. I understand what she meant. There is a difference between knowing that something existed and having evidence that it was alive. The skeleton gives you the first. The molecule gives you something closer to the second. A collagen sequence recovered from a T-Rex femur is not the animal, but it is something the animal made with its own cellular machinery, according to a genetic blueprint that encoded 66 million years of evolutionary history. And it has survived, improbably against every theoretical expectation, through the slow chemistry of iron and mineral and time, to be read by instruments in a 21st century laboratory by a doctoral student who got curious about what particle accelerators could see inside dense rock. That is an extraordinary chain of preservation and discovery, and it is one that, as of 2026, is no longer the story of a single laboratory's contested finding. It is the beginning of a field. The molecules are real. The methods are rigorous. The archive is vast. What remains is the work, patient, precise, cumulative, of reading what is written in the stone. The fossils were never only stone. We simply needed the right questions and the right instruments to hear what they had been holding all along.